Okay, this is part two of my series of presentations on proteins, and I have been having some technical difficulties. This is the third time I've tried this, and for some reason my screen recorder has just stopped working. So I am likely going to do these in installments of less than about 10 minutes or so that I don't keep wasting a ton of time. So the topic of this one is the nature of amino acids. So again, this is part two, and I'm even a little bit nervous to start the slide presentation because when I start the slide presentation, I cannot see that it's recording, which is that blinking red light up in the corner. So I think I'm just going to work from here. So an amino acid, regardless of what type it is, and there are 20, each amino acid has the same general formula, and that includes essential carbon, carboxyl group, an amino group, a hydrogen, and this is what's called the side chain. Okay, now I don't particularly like the way that this one is drawn. My preference would be to show the central carbon like this, my carboxyl group, COOH. That is the same group you saw when we talked about carboxylic acids. The amino group, which is the same group that we saw when we talked about amines. The hydrogen coming either up or down, it doesn't matter. And then the R group, which is the side chain coming either up or down. The main thing about the way I've represented this is that I have my carboxyl group on the right, and I have my amino group on the left. And if I continue to represent these in this way, it will help when we start talking about how amino acids come together to form proteins. So on the next one, the next slide is just a blow up of the same thing. One thing that I do want to point out is in this, again, I probably would have drawn this with my R group coming down like that and my amino group. The reason I have the N there is because the N is directly attached to the carbon and not the hydrogen. If I were to draw it on this side like that, I would have drawn NH2 like that. But since it's on the other side, I do want to bring it right to the nitrogen. So anyway, if this is a carboxylic acid and this is my number 1 carbon, this is the number 2 carbon, which is known as the alpha carbon. So that central carbon that we're always talking about with the amino acids is the alpha carbon. Now, one of the characteristics that occur with amino acids is that every single amino acid has what is called chirality. Actually, there's one that doesn't, but there's only one case that it doesn't. And chirality occurs when there are molecules or objects that are mirror images of one another. So if you imagine this as a plane, as a mirror plane, you can look and see that these are mirror images of one another. Your hands show chirality. So if you place your hands side by side with your palms down, and I hope that you're doing this right now, and you notice they're mirror images of one another, assuming that you have your thumbs and all of your fingers. So if you rotate them a little bit and have your palms facing each other, they're still mirror images of one another. But they're not the same, because if you had two left hands the function would be very, very different. So they each have slightly different functions, and they're able to do things in slightly different ways based on whatever you're trying to do. So chiral molecules are considered non-superimposable on each other. So if you have your hands facing, your palms facing down, and you take one hand and place it on top of the other hand, you'll notice that they don't line up. Your thumbs are on opposite ends, on opposite sides of each other. So even though they're mirror images of one another, they are not the same. So each amino acid has two different versions of the way it can exist, but I will tell you that the amino acids in the human body are the L version. And the L version, L stands for levulatory, and it is in general, in chemical terms, we refer to that as the left-handed isomer of that molecule. And D stands for dextrorotary, and that stands for the right-handed. And again, in the human body, it is only this left-handed version of the molecules that exist. So I have a series of pictures that show the nature of the chirality of amino acids. And you can see that these are mirror images of one another. And if I just twist them a little bit, you can see that these are still mirror images of one another. Now, it is only one of these variations that exists in the human body, and it's the left-handed version. And without looking it up immediately right now, I'm not really sure which of these represents the left-handed version. I might be able to figure it out if I look backwards. No, it doesn't really matter. So the nature of this, the purpose of this installation was just to talk about the nature of the amino acid, how every single amino acid has a specific form, that central carbon. I'm going to choose a different color here. That central carbon attached to a carboxylate group. On the other side, attached to an amino group, attached to a side chain, and then attached to a hydrogen. There is one amino acid in which the side chain is actually just another hydrogen atom. And I don't remember what it is off the top of my head right now, which is a good reason that you aren't going to have to memorize any of these either. So in the next presentation, I'm going to talk about the amino acids themselves and how we can classify them. And we will look at a model kit that looks like this. So that is the end of part 2, the nature of amino acids in our proteins series. And